The folate receptor-mediated detection staining solution is a novel technology for detecting neoplastic changes in the cervical epithelium. The solution contains folic acid conjugated with reduced methylene blue. FRD can be used as a primary screening tool for precancerous and cancerous lesions, in triage of HPV-positive patients, in triage of women with the result of ascus, or as a co-test with HPV and/or cytology testing. The principal mechanism of action is based on the overexpression of the folate receptors and the elevated rates of reactive oxygen species in tumor cells. When the FRD staining solution is applied to these cells, the folic acid-reduced methylene blue conjugate binds to the folate receptor on the cervical epithelium membrane and endocytosis occurs. The acidic microenvironment in the endosome causes dissociation of the folic acid and reduced methylene blue. Reduced methylene blue is released into the cytoplasm, where an oxidation-reduction reaction occurs. Catalyzing iron ions lead to the oxidation of the reduced methylene blue. The folate receptors recycle back to the cell membrane, and through osmosis, the oxidized methylene blue exits the cell, providing a visibly detectable result by way of color change. The folate receptor-mediated detection method is simple and straightforward. Start by placing the patient in a lithotomy position. Expose the cervix with the aid of a speculum. Use a dry gauze or cotton swab to gently clean the cervix of any secretion or exudate. Align the red mark support bar on the applicator with the 12 o'clock position of the ectocervix. Insert the applicator until it is covering the entire ectocervix. Press against the ectocervix for 10 seconds, ensuring complete contact between the ectocervix and the polyurethane foam. Next, push the plunger labeled with the letters FRD, enabling the tapered tip of the applicator to enter the cervical canal for sampling. Press the applicator continuously against the cervix for another 10 seconds. Finally, rotate the plunger to the right, locking the tapered tip in place. Press the applicator against the cervix for 5 more seconds. Carefully remove the applicator. Slowly dip the foam portion of the applicator into the FRD staining solution bottle. After complete absorption, or about 30 seconds, remove the applicator. Next, return the applicator to the plastic packaging. Insert the packaging with the applicator inside it into the testing slot on the side of the automated sample processing system. Enter the patient's information and click the Scan button. The test will begin and the result will print automatically within 90 seconds. Clinical studies have proven that FRD provides a sensitivity of CIN2+ to be 80%. Specificity is between 70 to 80%. Used in triage with cytology, the sensitivity is between 90 to 99% and improves overall detection. After sample collection, results are available within 90 seconds. FRD detects abnormal cervical lesions of both the ectocervix and the cervical canal. FRD has been evaluated for use in 5 continents in both renowned institutions and hard-to-reach areas. It has garnered great acceptance from the medical community.